So, welcome back. This is part 2. This is our applied section. So we're going to look at abdominal pain, vomiting, diarrhea, mucus, PR, bleeding. There's also a handout, and it's in the introduction to the module section, that's about the unwell person. I think I talked about it in respiratory, but it applies to this section as well. I'll remind you at the end to go and have a look at it again. So let's start with abdominal pain. We'll also then talk about tenesmus. So we have nociceptors on the mesentery and peritoneal surfaces, and these are mainly A delta fibers. We also have nociceptors on the mucosae and the smooth muscle of hollow organs, and these are mainly C fibers. So start with transduction. We have transient receptor potential, hopefully they sound familiar, channels that will obviously create little localized EPSPs that will get summated at the trigger zone and lead to action potentials. And these TRP channels can be transduced mechanically, chemically, or both. So these are polymodal. Mechanical stimulation is going to be due to things like distension, increased peristalsis, torsion, pressure, any mechanical changes. Chemical are going to be stimulated by histamine, serotonin, prostaglandins, bradykinin, and a few other inflammatory mediators. Plus, in addition to the TRPs, we also have purinergic receptors, which of course bind adenosine and ATP, and we have acid sensing ion channels, A6, that bind H+. In terms of the types of pain, we can define it as parietal or visceral. Parietal pain is sharp and well localized, so we know what type of fiber we're going to have here. It tends to be chemically stimulated, and it's carried by A delta fibers, which of course are myelinated and fast conducting. Hence, it's sharp. A delta fibers have primary afferents either side of the abdomen, and these have their own second order afferents. So therefore the sensory cortex can localize the source of the pain. So hence parietal pain is sharp and well localized. Visceral pain is a dull ache. It tends to be mechanically stimulated and carried by C fibers. C fibers, of course, are unmyelinated and slowly conducting. Hence, we have the dull ache. Another difference here is that the primary afferents on either side of the abdomen share a second order afferent. So they come round into the dorsal horn on each side and then synapse with the same second order afferent. So therefore, because of that, the sensory cortex has no idea which one transduced the pain. So it can't localize the source of the pain. So it's going to be very poorly localized, but it tends actually to be perceived as central. So these are people that come in with their hand across their umbilicus when you ask them to show them where the pain is. In parietal pain, they're more likely to point to a specific area. And of course, with C fibers, we have the issue of neurogenic inflammation. This causes sensitization peripherally and centrally, in acute conditions and has, in chronic conditions, the potential to develop into long-term potentiation of those synapses. So let's have a look at that in terms of a nice diagram. The grey bar in the middle is supposed to be the spinal cord, and there you can see the thalamus, and from the thalamus we have the tertiary or third order afferent which goes up to the somatosensory cortex. So let's look at A delta first. A delta fibers in the abdomen, as we said, tend to be chemosensitive. So we're talking about inflammatory mediators, things like that. They innervate the mesentery and peritoneal surfaces. They're myelinated and fast conducting, and therefore we get sharp pain. They also have their own second order afferent. Okay, that second order afferent crosses over to the contralateral spinothalamic tract to go up to the thalamus, but they're kept entirely separate. Therefore, the cortex knows which side the pain is coming from, so we get localized pain. Let's have a look at that in terms of C fibers. C fibers in the abdomen tend to be mechanosensitive. They innervate the mucosae and the smooth muscle of hollow organs. C fibers are unmyelinated, so therefore they're very slow conducting. Therefore we get dull pain. C fibers share a second order afferent. So in other words, what happens is the C fibers come into both dorsal horns, but they synapse with one second order afferent. So there's no way the cortex is going to know where it's coming from. So it tends to be perceived as central, or it's very poorly localized. Do have a look at Flaser and Goldberg. It's an article I've put on Moodle on abdominal pain. Tenesmus, this is the painful sensation of incomplete defecation. So a patient may say after they've been for a poo, it's a bit sore and a bit painful and they feel like they want to go again. There's mainly theories about this. But the theories are that it could be caused by tumor invasion of the lumbosacral plexus. And you can see there in the diagram, that plexus, those neurons are wrapped around the rectum. This causes neuropathic pain because it's actually damaging those neurons. Another source of this discomfort could be the activation of somatic afferents by any inflammation. And finally, it's theorized that smooth muscle contraction stimulates the autonomic afferents. And this is quite logical in terms of the patient going for a poo, and of course they're going to have smooth muscle contraction in order to do that. And that stimulates the autonomic afferents. Which brings us on to nausea and vomiting. Posh word for vomiting, of course, is emesis. The vomiting center itself lies in the medulla, alongside all the other centers, the cardiovascular centers, the respiratory centers, and whatever else. The vomiting center, although, takes input from other areas, and then it triggers the vomiting reflex. So it's sort of like the master of vomiting. In the diagram there, you can see it indicated as VC. In itself, it can be activated. It has receptors for histamine and acetylcholine and possibly a few other things. So it can be activated directly, But more often than not, there's 4 other areas that input into it. As we said, when it receives the input, it's the area that directly triggers the vomiting reflex. So our 4 areas. Just above it on the diagram in purple, you can see the CTZ, the chemoreceptor trigger zone. This is adjacent to the medulla, and it has receptors for things like opioids, some drugs, particularly chemotherapy drugs, alcohol, and endotoxins. When stimulated, it will then send action potentials to the vomiting center. The gastrointestinal tract, which interests us more here, the gastrointestinal tract has mechanoreceptors and chemoreceptors that transduce and then transmit via vagal afferents to the nucleus tractus solitarius, the NTS. You can see it there just above the vomiting center in blue. And the NTS then sends action potentials to the vomiting center. A couple of other areas, we have the vestibular region. This is responsible for motion sickness in Meniere's. It has receptors for histamine. Also higher centers of the cortex and via the thalamus. And this reports into the vomiting center and causes nausea and possibly vomiting when we're anxious, in significant pain, in an area where there's a noxious smell, sight, or taste. So these are the things that make us feel sick that we experience. And here's our areas all displayed out. On the left there you can see our gastrointestinal with its chemoreceptors, mechanoreceptors, serotonin receptors, but these of course fall under chemoreceptors anyway. And they're quite key because the GI tract produces most of the serotonin in the body. That reports in via the NTS to our vomiting center in the medulla. The vomiting center in the medulla sends back efferent action potentials to the gastrointestinal tract to cause vomiting. So let's start at the beginning with transduction. As we said, we can have mechanical stimulation of our, if you like, vomiting receptors. And this will be caused by distension due to perhaps reduced motility, reverse peristalsis that we'll talk about in a minute, because that's actually an outcome of the vomiting reflex as well, overloading or obstruction. So these sorts of mechanical changes. Chemical things, exogenous toxins. So bacterial toxins can stimulate vomiting. And in one case, we actually have a virus that secretes a toxin as well. Inflammatory mediators, and, as we said, serotonin. And as I said, 95% of it is produced in the gut. Normally the levels are regulated by the microbiota. There's another useful act of the microbiota. And serotonin regulates motility via the enteric nervous system, which we'll talk about later. Transmission, as we said, is via the vagus nerve up to the NTS. The NTS is just a column of sensory nuclei within the medulla. Via vagal afferents, though. That's all then relayed to the vomiting center. Which brings us on to the vomiting reflex itself. So, it starts with this retroperistalsis. This is also known as reverse peristalsis or antiperistalsis. Basically, what's happening is instead of pushing everything down, we start pushing it back up. and it can start from as far down as the ileum. The intestinal contents are pushed up into the duodenum and then into the stomach. The esophageal sphincters relax. The glottis closes after we take a deep breath because we need air, but also we need to protect the lungs from aspiration. The diaphragm and the abdominal muscles contract. And what this does is, of course, it's going to compress everything inside, including the stomach. So the stomach becomes compressed. This forces the contents that have come up through retroperistalsis, forces the contents up through these relaxed sphincters and the relaxed esophagus and out. In addition to this, there's some other things happen. If you think about the last time you vomited, you perhaps experienced some of these. Before you vomit, you get that water in your mouth. You'll get that increased salivation. It's called water brash. And that's stimulated by the parasympathetic nervous system. You also get sympathetic-driven pallor and diaphoresis. Pallor because the sympathetic nervous system, yes, it peripherally vasoconstricts, takes the blood away from the skin, so you go pale, and causes diaphoresis, sweating. The sympathetic nervous system, I would hope we know this by now, also increases heart rate and increases blood pressure, so increased contractility and all the rest of it. So these are additional signs that we get alongside vomiting. So another delightful topic for you. Let's talk about diarrhea. Diarrhea, what's happening is the intestinal fluid output is overwhelming our ability to reabsorb that fluid. So we might release a load of fluid in the ileum. By the time it gets to the colon where water is absorbed, there may be so much of it that the colon can't reabsorb it. So hence we have diarrhea. Two mechanisms that cause watery diarrhea are osmotic and secretory diarrhea. Osmotic diarrhea, as the name would suggest, is about creating hypertonicity in the intestinal lumen. Things that might do this would be us actually eating something that we can't absorb. So sorbitol is a classic example. Remember intracellular sorbitol when we talked about blurry eyes and diabetes? This is the sorbitol in sugar-free mints, things like that. If you eat a whole pack of them, you get lots of sorbitol in the lumen of the bowel, and that, of course, is going to draw water into the lumen of the bowel because it's increased the tonicity, increased the osmotic gradient. Or it could be due to malabsorption. This might be an endogenous abnormality, such as lactase deficiency. If you don't produce enough lactase, you can't absorb lactose. So you get lactose intolerance. That lactose will sit in the lumen of the ileum and it will draw water out by increasing the tonicity, giving hypertonicity. Another possible cause of malabsorption is damage to the enterocytes. If you damage the enterocytes of the villi, they're unable to absorb any solutes that they should be absorbing, so nutrients of various kinds, glucose, all that sort of thing. If they can't absorb it, it's going to remain in the lumen and cause hypertonicity. And damage is possible from inflammatory processes, so in inflammatory bowel disorders, or due to pathogen action, so bacteria and viruses destroying the enterocytes. And of course, this hypertonicity, as we said, in the lumen, is going to draw water in due to the osmotic gradient. greater than we can get rid of, than we can reabsorb when we get down to the colon. Before we talk about secretory diarrhea, we need to talk about the normal physiological regulation of fluid in the intestinal lumen. So, remember our crypts and our villi. The crypts were the deep bits at the bottom. Down there, the crypt enterocytes, if you remember in the crypt, they tend to be more secretory. And this is one of the things they secrete. The crypt enterocytes have these channels called cystic fibrosis transmembrane conductance regulators, shortened to CFTRs, thankfully. These channels are regulated via the activation of adenylate cyclase by a G protein. Oh, remember all of this? So, of course, if you activate adenylate cyclase, you get cyclic AMP. And if you remember, one of the things that cyclic AMP could then go on and do is activate protein kinase A. Protein kinase A phosphorylates. In this case, what it does, it phosphorylates our CFTRs and makes them more likely to open and release their ions. And what they release are chlorine primarily, and to a lesser extent some bicarbonate. So of course this efflux of chlorine and bicarbonate, these are both anions. Chlorine is an anion, our bicarbonate also carries a negative charge, but it's a molecule. So these are released from the enterocytes into the crypt of the intestinal lumen. Now, because they're negative, sodium wants to follow them because, of course, we've got an electrical gradient going on, opposites attract. So sodium follows this electrical gradient created by the chlorine and the bicarbonate and enters the lumen through our selectively permeable tight junctions. And these, of course, are between the crypt enterocytes. So we've got chlorine and bicarbonate coming out of the enterocytes being effluxed, and we've got sodium coming through the tight junctions between the enterocytes. So we've now got a lovely little soup, if you like, of ions. The combinations of this creates an osmotic gradient within the crypt. This draws water into the lumen, which also comes through the tight junctions. So this is normal regulation of fluid. We always need some fluid in the lumen of the ileum because we need to mix everything up when it's quite mushy there. So we always need some fluid. So we need this process to happen to draw water into the lumen. The ions are then, before they're lost down through peristalsis, the ions are reabsorbed by the enterocytes at the top of the villi. Remember, top of the villi, the job is absorption. Crypts, the job is secretion. So the ions are reabsorbed, but of course the water mixes in with the contents of the ileum. The CFTRs are so named because they occur in the lungs and other places. And what their role there is to help keep mucus moist, provide the water content of mucus, the soul, if you remember, layer. And of course, in cystic fibrosis, hence the name, there is a mutation involving the CFTR gene. So therefore, you don't get nice moist mucus, so you get mucus plugs. So let's again have a look at another diagram. So here we have the ileum, we have our crypt enterocytes at the bottom, and in them we have our CFTRs. Between them we have our tight junctions. So first of all, we have chlorine and to a lesser extent bicarbonate effluxed from the CFTRs. Sodium follows, but that comes up between the cells through the tight junctions. And of course, what follows all of that is water. Our ions there will eventually be reabsorbed at the top of the villi, but the water will contribute to keep the contents of the ileum at the right viscosity, if you like. So, having talked about normal physiological fluid regulation, we can now talk about secretory diarrhea. So think about it. If the ions secreted by the CFTR channels and via the tight junctions, so we have our chlorine bicarbonate via the CFTRs, sodium via the tight junctions, if these are increased, what would happen? Yes, what happens is more water comes in. So in secretory diarrhea, what happens is adenylate cyclase is stimulated. Of course, this is going to give us increased synthesis of cyclic AMP, which is going to activate more protein kinase A. So therefore, we're going to have phosphorylation of CFTRs. This sensitizes the CFTRs and they increase their chlorine and their bicarbonate influx. Increased chlorine and bicarbonate, of course, increases the amount of sodium that follows the electrical gradient, comes up through the tight junctions. And the increased osmotic gradient of this soup of ions increases the water movement into the lumen. In addition, if on top of that you've got damaged villus enterocytes, the absorptive ones, they're going to reduce their reabsorption of sodium and chlorine and bicarbonate at the top of the villi. So that is going to sit in the lumen then and draw even more water out. Things that do this, that upregulate the adenylate cyclase, the cyclic AMP, and the phosphorylation of CFTRs, are things like bacterial toxins. So these bacterial toxins, examples are cholera, E. coli, or salmonella. Our virus that does this, that produces toxins, it's really one of the very few, if not the only virus known to secrete toxins, is norovirus. Some laxatives, this is how they work, they upregulate the CFTR channels, and some other drugs. Plus there's a few other things. Arsenic, not that you're going to go taking that. Insecticides, all those, but caffeine does. So caffeine basically will increase the fluid going into the lumen of your GI tract and increase your ability to poo. And this is a delightful picture of rice water stools in cholera. Luckily not something we get an awful lot of in the UK, but in cholera someone actually passed that. Now that is really watery diarrhea. Our other problem in terms of diarrhea is the immune response itself, of course. So cytokines, proteases could potentially cause enterocyte damage, apoptosis, and necrosis. This is going to cause malabsorption. It's also going to give us vasoactive changes, which is going to cause exudate, adding to all of this fluid, and possibly upregulate the goblet cells, so we get increased mucin, combined with the increased exudate, so we get mucus. There's also the potential, as we said before, for damage to the tight junctions. So various bacteria good old norovirus again, and TNF-alpha disrupt the tight junctions. This, of course, is going to have multiple effects. We've got increased water and sodium getting transported into the lumen because it's not being regulated by the tight junctions. We've also got the passage of exudate into the lumen. And because, remember, one of the jobs of tight junctions is to maintain apical basal polarity to stop those little transporters and channels and everything else moving around the side of the enterocyte, we lose that apical basal polarity. And the potential is that's going to have an impact on the enterocyte's ability to absorb anything in the lumen. And of course, we get the loss of the solutes because they go between the enterocytes. We can't then process them within the enterocyte so that we can then use them. And of course, a loss of the barrier against pathogens. If you remember, we looked at that before. Pathogens can get between the enterocytes. They can get into the subepithelial tissues where they're harder to find and kill. I promised you a bit more on serotonin. And serotonin has quite an impact in terms of diarrhea. I mentioned also the enteric nervous system. Now, the enteric nervous system is sort of a separate nervous system outside of the central nervous system that regulates motility and it regulates iron secretion from those CFTRs. Our enterochromaffin cells, ECs within the epithelium, release serotonin. Remember, we said 95% is produced in the gut. They release it downwards into the underlying connective tissue. Once it's there, it activates the neurons of the enteric nervous system. Activation of the enteric nervous system triggers the release of other neurotransmitters such as ACH and substance P, which all excite peristaltic and secretory reflexes. So not only have we increased fluid, we've got increased gut motility pushing it out. And some bacteria and viruses use this mechanism. They stimulate serotonin release from the ECs, the enterochromaffin cells, which of course is going to then increase diarrhea. Excess serotonin increases peristalsis, as we said, and it also increases the iron secretion from the CFTRs and consequently the water moving into the lumen. Serotonin, if you remember, we had receptors for serotonin in terms of our vomiting receptors. So it stimulates vomiting. It also can bind to nociceptors and stimulate pain. And not surprisingly, really, when you look at those symptoms, abnormal serotonin signaling has been implicated in IBS, irritable bowel syndrome. And what they noticed was that SSRIs, serotonin reuptake inhibitors, have improved the symptoms in patients who take it who happen to have IBS. So a quick word on antibiotic-associated diarrhea. In patients with primarily non-GI infections, diarrhea occurs in 5 to 25% of those receiving antibiotics, particularly the broad-spectrum antibiotics. And of course, what they're going to do is they're going to disrupt that microbiome, that delicate balance in terms of the commensal organisms within the GI tract. as a result, what could happen is that, remember I said that these pathogens can be really helpful. So they tend to help us metabolize carbohydrates so that we can absorb them. If we don't have those commensal microorganisms to do that, then those high molecular weight carbohydrates don't get absorbed. If they don't get absorbed, they're going to sit in the lumen and they're going to cause osmotic diarrhea. Bile acids are another key thing. They tend to be metabolized by the microbiota. In this case, because the microbiota is a bit destroyed, they're not metabolized, so they tend to sit in the lumen. But what that also does, the bile acids stimulate, they upregulate adenylate cyclase, cyclic AMP, etc., and therefore upregulate the CFTRs. So those unmetabolized bile acids increase the chlorine efflux from the CFTRs. Of course, sodium's going to follow, water's going to follow. And of course, the other role of the microbiota is to compete with pathogenic bacteria and prevent their colonization. So without them, we get overgrowth of pathogenic bacteria. And up to 20% of antibiotic-associated diarrhea cases have Clostridium. They're due to Clostridium. Of course, Candida overgrowth, everyone talks about, oh, I'll get, yeah, Candida overgrowth. That could happen, but it doesn't seem to have an impact on diarrhea. Okay, so I'm not going to talk too much about mucus because we covered a lot about its secretion and its upregulation in respiratory. So you need to go back to that, and then it applies in the same case, but of course we don't have submucosal glands, we're just talking goblet cells. So have a look at the respiratory lecture for the full details of goblet cell upregulation. This, of course, gives us increased mucin secretion, so it's going to make the mucus stickier. As I said, other than the esophagus and the duodenum, mucin is supplied by goblet cells only. In the ileum, mucus is quite important because it protects the enterocytes from pathogenic organisms. Remember those pattern recognition receptors, antimicrobial proteins, etc. The colon, as we said, has a double layer because here what we really want is lubrication. So the lowest layer is attached to the epithelium, and then you get this outer loose layer containing the microbiota, the non-pathogenic commensal organisms that can slide along the top of that. This helps to lubricate the passage of the semi-solid and solid feces. Mucus upregulation and exudate, what I really should say there is mucin upregulation and exudate, will occur in any inflammatory or infective condition. The combination of mucin upregulation and exudate gives us increased mucus. So anything that causes an inflammation in the ileum, ileitis, or in the colon, colitis, will give us mucus upregulation. PR bleeding, I should imagine you should be able to work this out for yourself by now, because this is usually stimulated in pathogenic conditions by angiogenesis. Because we have inflammation, we have vascular endothelial growth factor and other growth factors, fibroblast growth factors, remember, all stimulate angiogenesis. So angiogenesis is possible in malignancy to support tumor growth and metastasis, chronic inflammatory conditions due to concurrent damage and repair, or in acute infection and inflammation due to enterocyte or mucosal damage. And in acute conditions, this is what is generally referred to as dysentery. In the descending colon and rectum, you're going to get a gross bleed and blood-streaked stools. You can imagine we've got this formed lump coming down. It's going to rub against these delicate little buds developing from capillaries and break the top off them. And you're going to get a coating of blood on the stool. Or, if it's a massive capillary that the inflammation has broken into, you could get a larger bleed. The ileum and the proximal colon, of course, it's more likely to be fecal occult blood because there we've got liquid. So the blood will mix with the liquid. You won't necessarily see it. If there's enough of it, it will make it all that black and sticky. But in small amounts, you won't necessarily see it, hence the word occult. The other thing to remember is that if someone has persistent PR bleeding, there is a potential for anemia. Cachexia and fatigue. We have various issues here that relate particularly to GI conditions. And this could be malabsorption, couldn't it? Because we've got damage to enterocytes. Or if you've got regular diarrhea, it's hurrying all the contents through rather quickly, too quickly, to be absorbed by the enterocytes. So we have the potential here of malabsorption. This, of course, could compromise multiple systems because what we're not absorbing could involve various vitamins that are vital to the synthesis of a particular enzyme, for example. So lots and lots of systems involved in addition to reducing energy because we reduce ATP synthesis. As a result, we tend to get alternative sources such as stored fats, carbohydrates, and eventually proteins get broken down and used as energy. This, of course, is going to deplete body stores. However, there are other mechanisms that are common to so many chronic conditions, chronic inflammatory conditions. The release of cytokines have an impact in multiple ways that are all detailed in that unwell person handout. So other mechanisms will contribute to any acute or chronic condition that involves inflammation and cytokines. As I said, if you've not already, please make sure that you have read and understood the Unwell Person handout. This is in the module preparation section because it encompasses so many different systems, so many different pathologies that I couldn't really allocate it to one single one. So it's in the module preparation section. If you haven't read it, please go back and do. And that brings us to the end of our gastrointestinal topic. As I said, please complete all the independent learning activities and do all your usual things to consolidate and learn this topic before we move on to the next one.